Kia ora koutou, ko Sue Brewster taku ingoa. Well, thank you everybody for coming this evening, and we just probably have a few more people roll in, and of course we've got quite a few people joining us online tonight as well. So as I self-introduced, my name is Sue Brewster, and I possibly had the best job in the world for the last 8 years as Executive Director of Auckland Medical Research Foundation. So I know all of you here tonight and those joining us online are going to be super excited to hear from our two internationally acclaimed professors, Professor Robert Gourdie and Professor Donna Rose Addis. So welcome. But look, I thought I'd kick off tonight with a karakia. And this karakia speaks to seeking knowledge for understanding and having purpose in all we do, which is perfect for the essence of tonight. E te hui. Whaia te mataranga kia marama, kia whai taka nei, mahi katoa, tu maia, tu kaha, araha atu, araha mai, tātou ia, tātou katoa. As always, I have some quick health and safety instructions, but most of you probably know that if there's an emergency, we will go up the steps and out the front to the grass area. We'll wait there until further instruction. Bathrooms are up there to either side off the main foyer. Not that that's a health and safety issue, but you never know. It could become one, couldn't it? So just a little bit about Auckland Medical Research Foundation. For those of you that don't know, we are actually celebrating our 70th year anniversary. So this is the second of our events for this speaker series, and we've had 3 before this as well. So, but very privileged to have both professors come back from overseas. So we also had wonderful 70th year partners join us, and we'd like to thank the University of Auckland as our host tonight and as one of our 70th year partners, also the Auckland University of Technology and JMI Wealth, who are now, were JMI Wealth, but they're now Shaw and partners. So, and this is also the celebration of awarding over $100 million worth of medical research funding. So, you know, which is a pretty phenomenal achievement since 1955. We are the largest independent funder of medical research across the spectrum of all disorders and diseases, and that's in Aotearoa, New Zealand. So, and we have gained a really great reputation for our steadfast governance and our amazing medical committee, some of who we have here tonight. And they obviously provide a really robust application process and review process for everybody for our funding. So, sadly, I'd like to be able to say that we can fund all of the eligible applications, but we can't, so we fund less than 20%. However, the 20% that we are able to fund, we give about $4 to $4.5 million funding every year, and our board, in times of diminishing support for medical research, have actually said that's going to be a minimum, an annual minimum. So we're incredibly grateful to have a very supportive board and ones that know where the need is. So a big thank you to all of our donors too who help support us. And also for those of you that don't know, 100% of all donations go directly to our purpose, which is to fund world-class medical research, and that is because we have an external benefactor who actually provides all of our operating expenses. So once again, a really unique proposition as a charity here in New Zealand. So that's a glimpse of our foundation since our origins in 1955, and now on to tonight. So tonight, how we're going to rock and roll is we're going to have Professor Robert Gourdie up first presenting for us, and then we'll have time at the end of the presentation. That should be 40 to 45 minutes. Sound about right, Rob? Yep. And then we'll have time for question and answers. And then we'll have a really short break of about 5 minutes so you can stretch your legs, take a bathroom break, and we'll kick back off just after 7 o'clock. And that's for our people online to know that as well. But before I start our first introduction for this evening, I'd just like to thank the Ted and Molly Carr Medical Travel Fund because they made it possible for Professor Robert Gourdie and Professor Donna Rose Addis to come back to New Zealand for these. So we're incredibly grateful to have such two acclaimed professors back for our 70th anniversary. So, my first introduction of the night is Professor Robert Gourdie, a New Zealand-educated scientist and internationally acclaimed cell biologist specialising in cardiovascular and biotech innovations. Robert has worked in the United States since the late 1980s, and his research spans from fundamental cell biology to therapies moving towards the clinic. His lab has led promising treatments for wound healing, heart protection and cancer, and Robert and his team have pioneered technology turning milk exosomes into an almost ideal oral drug delivery system. Robert is now studying his own alpha-CT1 peptide for heart attack protection and as a radiation mitigating drug for cancer patients. Robert completed his undergraduate and graduate studies in Aotearoa, New Zealand, yay, and before doing his postdoctoral research at University College London. He is the Hayward Fralin Professor at Virginia Tech and Director of the Centre for Vascular and Heart Research at the Fralin Biomedical Research Institute at Virginia Tech. It's quite a mouthful, but this is how much work Rob does. He also founded ZQuil Bio Inc. and is the founder of the Tiny Cargo Company. But the very best person to tell you about his research tonight is Professor Robert Gourdie, and I'd like you to give him a very warm welcome.
Hi everyone, and thanks for coming out tonight. And thanks so much, Sue, for that very kind and very warm introduction. I'm not actually going to be talking about milk exosomes tonight. I'm going to be talking about my lab's work on electrical activation, so just heads up. All right, so this is the view off my back porch, the Appalachian Mountains. Very beautiful. It's not really the view off my back porch, but it looks something like this. We have a very nice view. We live on a mountaintop. And this is a slide I put up. I debated whether I put it up or not because there's a little bit of a joke embedded in it. And it's this. I put the slide up for my wife and she said, 22 hours from Charlotte, just like Roanoke. And the joke is, of course, Roanoke is 2 to 3 hours drive from Charlotte, but often the flights are cancelled between Charlotte and Roanoke, so you end up there overnight, and so hence the 22 hours. Anyway, joke is never good if you have to explain it. This is the Fralin Biomedical Research Institute where I work, and just a couple of things to notice. This trim around the edge of the institute is actually characteristic of nearly all the buildings at Virginia Tech where I work. It's called Hokie stone, and it's a particular type of limestone that has been used, as I mentioned, for most of the buildings at Virginia Tech. The boss wanted to originally have the entire institute built out of Hokie stone, but then he found out how much it cost, and he thought, well, a nice little bit round the bottom would probably work for us. Here's some disclosures. I'm not actually going to talk about any work that I'm in conflict with tonight because I'm going to focus more on our fundamental work in cardiac bioelectricity. All right, so first up, I just really want to thank and acknowledge all the members of my team. They're the ones that actually do the work. You'll see we've got many undergraduate researchers and medical students in the lab. And that's one of the aspects of the Institute. We're allied with a medical school, and the part of their 4-year degree, the medical students actually have to do research in a research laboratory as part of the requirements for the degree. So, and we've seen some very good ones over the years. All right, so here's a functional MRI of a heart. And what you're observing, of course, is the waves of electrical activation that the heart generates, prompting the mechanical pump action of the heart that's necessary to pump the blood around the body. Now, sometimes this very steady rhythm goes wrong, and we get an arrhythmia. And it turns out that really for the last 20 or 30 years, there's been very little progress made on developing new drugs and therapies to counter arrhythmias. And one of the barriers to development of new drugs for arrhythmia is the fact that we really don't have a particularly grounded understanding of how the heart electrical activation works yet. And I'll explain a little bit more about that as I go on. It's partly my opinion, but I think as you'll see, as I'll try and make the case, particularly as it relates to the conduction of the action potential in the heart, you'll see what I'm getting at. And of course, arrhythmias cause a large fraction of heart disease, stroke and disability. And here's some numbers. So sudden death and arrhythmias account for somewhere between 15 to 20% of all global deaths, with atrial arrhythmias accounting for around 3% of this number, 3% of the within the 15 to 20%. Atrial arrhythmias are often viewed as benign, but they're not. They're a major cause of stroke, for example, with blood clots forming in the atria and migrating to the brain. And as I've mentioned, safe and effective arrhythmia therapies remain limited. Another reason for this limitation is that pharma companies are often hesitant to invest in new drugs due to the high costs associated with development of these drugs, and also the risks. And one quite well-known, perhaps even infamous trial was a study called the CAST trial, in which the researchers looked at about 1,700 patients with clinically judged arrhythmias, and found that even though these sodium channel blockers that they were on did decrease the frequency of arrhythmias, they actually increased the rate at which the patients were dying, which, is not a good combination. All right. So what we're seeing here, and yay, the video's running, is the normal sequence of activation of the heart. And so, of course, this happens once every second or so. And for the average human, this will occur some 2 to 3 billion times over their lifespan. And, it's fairly relentless and reliable, and it has to be, because if you get something like this, a ventricular arrhythmia where the patterns of electrical activity become chaotic, unless it's converted back into a normal sequence of activation, you're dead. So, obviously something that one wants to avoid. So, what are the structures that underpin this electrical activation? So this is a cartoon of the ventricle, and you can see that it's made up of these brick-like cells, ventricular myocytes. They're connected together by intercalated discs, which are kind of represented. They're kind of hard to see on this. But if we look at an individual myocyte, and in this case we have an individual ventricular myocyte that's been stained with a dye that fluoresces in response to calcium, and put a recording electrode on it, we of course would record an action potential as the propagating wave propagates over the recording electrode. The first part of the electrical activation is caused by the sodium channels, which initiate this upward stroke in the activation phase. But then subsequently there are other populations of ion channels that contribute to the characteristic morphology of the action potential that we see. Okay, so you've probably heard me say the word cardiac conduction more than once. So one conception of cardiac conduction, i.e. the propagating wave of electricity through the heart, is as follows. So if we activate a cell, one myocyte, we of course generate an action potential, as I've just illustrated in the previous slide. And then it's conceived that the propagation occurs by the movement of charged ions through these structures called gap junctions, which sit between the cells, and generating the action potential in the downstream cell. Now, this, of course, is in direct contrast to the way most electrical activation occurs in the nervous system, where it's conceived that the action potential jumps from one neuron to the next at the neural synapse across this very narrow gap of extracellular space, around 100 nanometers in width. And so for 50 or 60 years, this has been conceived as the fundamental difference between the way that electrical activation conducts from one cell to another in the heart and in the nervous system. There are gap junctions in the nervous system as well, the so-called electrical synapses, but I'm just going to omit those for now, just to make this contrast. Okay, but it's complicated. So one of the pioneers in this field, and someone who started to raise questions back in the 1960s and 70s, was a guy called Nick Spiroulakis. And he made the observation that in avian hearts, the numbers of gap junctions connecting myocytes together is about 1,000th of the numbers that are found in mammals. And of course, birds have a pretty good cardiovascular system, right? You can see geese flying over the Himalayas, etc. Hummingbirds whose hearts beat at 1,000 beats per minute, yet they're doing it with just a fraction of the gap junction channels that are apparently necessary in myocytes. Another observation that occurred in the mid-90s was that connexin 43, the main gap junction protein found in the ventricle of mammals, and it accounts for about 99.9% of all connexins expressed in the ventricle. When it was knocked out, the mice were born live, but their hearts still showed normal conduction, again suggesting the concept of, are gap junctions really necessary for the propagation of activation from cell to cell in the heart? And then another couple of papers, really led by David Gutstein in the early 2000s, where they did cardiac-specific knockout of connexin 43. And in this case, the mice lived to around 4 to 6 weeks after birth. And when he looked inside, looked at where you'd expect to find the gap junctions in the intercalated disc between these cells, there was no evidence of gap junctions. And he collaborated with a group at Columbia, and they did double patch clamping of gap junctional conductance between cell pairs, and found that the level of conductance was theoretically too low to support conduction. And this is kind of the coup de grace from my point of view of raising questions about the conventional wisdom. There are dominant negative mutations of connexin 43 in humans. They're called ODDD. It's oculodental digital dysplasia. There you go, Sue. I got through those syllables.
Can you give a turn and point at the screen, please?
Sure.
Thank you.
Although this is in the way.
Smile for the camera.
Oh, I see. Yeah, oh dear.
Thank you.
All right.
Oh, you can step back.
Yeah, I'm going to step back so I can see what's going on the screen here. So, dominant negative connexin 43 mutations. Some of the proteins with these dominant negative mutations are actually trafficked normally to the gap junctions, and once they get mixed into the channels that form the gap junctions, they basically render the channel dead. And so you've basically got a gap junction which is fundamentally just a staple between cells and completely unable to communicate. And yet, as we can see here, here's an individual with one of these dominant negative mutations, and he's clearly survived to the point where, into adulthood. So, so there are big questions about, at least in the minds of some, about the role of the intercellular gap junction and the communication between cells in the heart with respect to action potential propagation. So where did I come at it from? Well, so here's a model of the gap junction. And it's a cluster of intercellular channels linking the cytoplasm of two cells. Here's a cutaway view of one of these channels. You can see it forms a kind of conduit. And, it was convenient to imagine that charge would flow through these channels and underpin the propagation of action potential from cell to cell. Now, my research over the years hasn't really focused on the gap junction per se, but we've focused a good deal on the edge of the gap junction in a region that we called the perinexus. And here, the undocked hemichannels which will eventually flow into the gap junction and dock with channels from the neighbouring cell to form these conduits, are actually held in place by a scaffold that includes this molecule here, zonular occludens 1, which in turn is linked to the actin cytoskeleton. And the ZO1 we found acts as a kind of brake for the hemichannel recruitment into the gap junction, controlling the rate at which the aggregation occurs during the life cycle of the gap junction. And we published a number of papers on this over the years. And I'll just, where's Matt? There he is. This is one of my graduate students from around 2008 to 2012, Matt Redd, and Matt and I gave the Perinexus its name. Now, one of the questions that came up as we started to understand the dynamics of this region at the edge of the gap junctions was, well, if it's a specialised subdomain, what else is going on there? And so we were curious to know what other proteins and structures might be present. So this is a GeoLink polymerization ligation assay in which we're looking at the protein-protein interaction signal in red between connexin 43 and NAV1.5, which is the voltage-gated sodium channel. And we've double-labeled it with connexin 43 immunohistochemistry in green, and you can see that the sodium channel interacting with connexin 43 is beautifully arrayed around the edge of the gap junction in this region that we refer to the perinexus. And so you can imagine these sodium channels are not actually sitting in the gap junction proper, they're sitting in the membrane cleft where the two membranes separate. If I can be so bold, they're sitting over an almost synapse-like domain, a domain that's separated by really less than 20 to 30 nanometers. So the voltage-gated sodium channel was present. In that same paper in 2012, we speculated that maybe, this is an old idea, we were sort of echoing Spirillacus, but basically starting to put the framework of a structure around Spirillacus's idea. Maybe action potentials weren't being directly transferred by charge moving between the cells, maybe they were jumping electrically across this very narrow gap of extracellular space where the sodium channels were located. So how might that happen? Well, here's a kind of model representation of two myocytes abutting in an intercalated disc-like zone. And we can see a couple of big old channels here representing the gap junctions. And then within the cleft of the extracellular space, these crosses represent sodium ions. So as the upstream cell depolarizes, the sodium ions move in from this very narrow gap of extracellular space into the first cell. This results, as the sodium is depleted in this space, in a change in the membrane potential within the space with respect to the upstream cell. This change in membrane potential then activates the sodium channels in the downstream cell, thereby underpinning the propagation. So you can see how in this more indirect step-like sequence, the activation may be able to propagate from one cell to the next. Of course, at this point, this is a conceptual, hypothetical proposal. Okay, and the type of conduction that's going on here is referred to as ephaptic conduction. It's a hard word to pronounce. It took me years to figure out how to say it properly. But it's Greek, of course, and it means touching. And in order for ephaptic conduction to occur, there has to be a high density of the sodium channels, and also you need to have a very narrow separation, an intermembrane width of less than 30 nanometers. And of course, these are features that are embodied in the perinexus, which, as I've just shown you, does have high concentrations of sodium channels, and as I'll demonstrate in a minute, does have the ultrastructure that would correspond to the very narrow separation that would be required. Now, this is Cy Veeragarvan. Cy was my postdoc between 2012 and 2015. You can see he's had a meteoric rise. He's now a full professor at Ohio State University, so he's done very well. He's a brilliant young guy. But he really sort of laid the groundwork for the structure that emerged and the function that we hypothesized might be underpinning of haptic induction. So here's a pair of myocytes, and you can see this light region here represents the intercalated discs that we find these electromechanical junctions coupling these two cells. And if we use the electron microscope, we can see this step-like structure that's hinted at by these white lines here is made up of sequences of different junction types. So on the treads of the steps, this is where the gap junctions tend to dominate. And then on the rises of the steps, we find adhesion junctions. The rough designation between these two zones is what's called the interplicate zone on the steps and the plicate zone on the rises. And there's just a labelling of the various structures, including the perinexus at the very edge of the gap junction structure we show here. And here's a higher magnification view of the perinexus. I've coloured it in red. It doesn't appear red on the electron microscope, of course, just to illustrate, again, this synapse-like cleft occurring at the edge of the gap junction. Okay, so I showed you some geolink data. But we also followed it up by looking using super resolution microscopy, which enables us to get a resolution down below 100 nanometers or so. And so here you can see connexin 43 in association with the voltage-gated sodium channel NaV1.5. And clearly there's a very intimate side-by-side association of these two populations of proteins. Now, the voltage-gated sodium channel is actually a complex. It has a pore-forming unit that's made up of this large protein called NaV1.5, but it also has an auxiliary or accessory subunit called the beta subunit. And the beta subunit is fascinating because it has an immunoglobulin domain, an immunoglobulin adhesion domain. And so one of the questions I asked myself when I realized this was, what's an ion channel doing with an adhesion domain? So we wanted to learn a bit more about it. Laurie Isom has done some very fine work on the beta subunit of the voltage-gated sodium channel over the years, but we decided that we'd raise our own antibody against the beta subunit because back 10 years or so ago, such reagents weren't widely available. And characterised it. Here you can see it's very beautifully localised with an intercalated discs and ventricular myocardium. We used beta-1. The gene for beta-1 is SCN1B, and we had SCN1B overexpressing cells, and it turned up at the right molecular mass within these cells. And we also had beta-1 knockout mice and could show that there was a loss of this 37 kilodalton band. So the antibody looked good, and we were happy it was working well once we had done the quality control checks. And we asked ourselves the question, where are we seeing it? So this is what we call an en-face view of an intercalated disc. And the way to imagine it is, if this is your myocyte, basically we're looking face on at the intercalated disc. And so, what we observed was that there was a very distinct partitioning of the beta-1 within the interplicate region. So here's N-cadherin, and you can see it's densely populated within the plicate region, but the corresponding interplicate region is very rich in this beta-1 subunit. And of course, as I just mentioned, this is where the gap junction and the perinexal structures that we're interested in are located. Of course, we did some super resolution on this as well, and this is probably the most distinctive one to look at. But here we can see super resolve beta-1 subunits associated with connexin 43, again in this very intimate side-by-side relationship. We actually developed some software for undertaking quantification on super res images, and we convinced ourselves that the beta-1 localizes at these interplicate domains, and is enriched within the gap junction perinuclei. So basically you've got this adhesion molecule in the zone of very close apposition between the membranes of two cells at the edge of the gap junction. So this is the kind of model that we started to build up around the gap junction. We have the gap junction proper itself with its docked hemichannels forming the conduits between the cells, but adjacent the gap junction we have the perinexus. And within the perinexus we've got undocked hemichannels of connexin 43, and these interact with the voltage-gated sodium channels. And the extra bit of information is that we also have beta 1. And we hypothesized that the beta 1 was associated in these homophilic interactions across the very narrow gap of extracellular space, which actually served to align the sodium channels across this space so that they become almost junction-like themselves. And I'll talk a little bit more about that in a little while. Okay, so within the perinexus we have the requirements of a phaptic nanodomain. Cell membranes are separated by 30 nanometers. There's a high concentration of sodium channels. And do I have an extra one there? And of course, we hypothesize that the sodium channels in perinexus is stabilized by this beta 1 mediated adhesion. Okay, so one of the things that my lab specializes in doing is making peptides against interesting domains of proteins that we're interested in. And so here's a 3D model of the beta 1 subunit. And so we identified the prospective adhesion surface of the immunoglobulin. Goodness, what happened?
Blank slide. Just try pressing the next one.
Okay, so we made a peptide corresponding to the adhesion surface of the beta 1 immunoglobulin domain, with the idea that we'd glom the peptide onto the immunoglobulin domain and disrupt these homophilic interactions between the immunoglobulin subunits. And it's going to go blank again. All right. We also proposed, and this was in a review from a few years ago, and here we can see the cleft of the perinexus. Here's the gap junction, and here's where the two membranes separate. That these two the homophilic interaction of adherent beta subunits would very nicely fit into the dimensions of this gap. And of course, the idea was that we would use our peptide, which we call BADP1. I didn't name it, Cy did. You know, I don't think it's a particularly great name, but you know, he liked it. It amused him. To disrupt this interaction. All right. This was some of the initial characterization we did. We generated a number of control peptides, including a scrambled control peptide, and also peptides based on mutations that occur in humans within this peptide sequence. And here's one of them, R85D. And we also used a technique called electric cell substrate impedance spectroscopy. And this is essentially an assay that uses electrical current flow from a multi-point electrode to assess the level of adhesion that's occurring between cells. And so when we put the peptide onto cells that were overexpressing the beta 1 subunit, we saw a dose-dependent loss of the adhesion between the cells. And this adhesion loss was less dramatic in the case of the peptide with the mutation, and also not significantly, even though it's not showed here, there was no significant loss of adhesion in response to the scrambled peptide. Another thing we wanted to know before we actually got into animals was whether the peptide would have direct effects on the electrical characteristics of the myocytes. Obviously, we wanted to target the adhesion properties of the beta 1 subunit, not its effect on the channel itself. And so we worked with a colleague at Ohio State, Isabelle Deschênes, and she did some single patch clamp studies, or single myocyte patch clamp studies of the sodium currents in these cells and also in the action potentials in these cells at different concentrations. As far as we could tell, there was no effect on the sodium activation properties or the action potentials. So that was good. So the next thing was to actually assess the effects on adhesion. So here's a different type of representation of the beta-1 subunit. Here's the BAD-P1 peptide that corresponds to the, as I mentioned, the adhesion surface of the immunoglobulin domain. And we perfused it into live beating guinea pig hearts at around 50 to 100 micromoles. And what we found was that indeed the perinexi were blown out. So here's a controlled perinexus, and you can see it's maintained this beautiful spacing. Again, the yellow coloration is me. Yeah. But there's a clear loss of adhesion within the perinexus itself, a dehiscence of the proximity between the two membranes. And we measured it, and it was significant. And if we looked at other parts of the intercalated disc, the BAD-P1 didn't affect the intermembrane spacing in those regions. We were required by the reviewers to look in the beta-1 knockout mouse to see whether the loss of the beta-1 subunit resulted in a similar phenotype to the peptide, and indeed it did. And we got a pretty dramatic increase in the intermembrane spacing in the perinexus in these animals. So that was a helpful comment from a reviewer. Added into the paper. We also sent the peptide to Imperial College, to our colleague Julia Gorolich, and Julia is able to basically interrogate different regions of membranes of myocytes using fairly sophisticated patch clamp technique called SICM. And she compared the response to the cells on the lateral surfaces, but also in the junctional domains. We also sent her a GFP adenovirus that expressed GFP tagged to connexin 43, so she could position her patch pipettes very close to the gap junctions. And what happened in response to the peptide is we got this time and also dose-dependent loss of the junction-associated sodium currents. And then finally, we're back in the guinea pig heart again, the isolated guinea pig heart, and we had electrodes on recording the electrical activity. And what we find is with increasing doses of the peptide, we get an increase in the ventricular tachycardia characteristic of arrhythmias in these hearts. So what I've shown you so far is that we have the structure, the perinexus, it's rich in sodium channels, and these sodium channels appear to be adherent across the perinexal space, and if we disrupt that adherence, bad things happen to the sodium channels and also the heart rhythm. This is my colleague, Steve Pulsing, at the Institute. Steve and I actually recruited Steve to the Institute. That's a long story, I won't go into it. But nonetheless, we're friends and have been collaborators now for the last dozen or so years. And Steve has really gone on to look at this de-adhesion of the perinexus in various scenarios, including, importantly, in disease scenarios. He's obtained tissues from human patients with persistent atrial fibrillation and found that significant de-adhesion can be found in these patients, and published some very influential studies over the years, including this one in Circulation Research that just came out a couple of years ago now. One of the criticisms that we, first of all, when we started talking about apoptic induction in the heart, people said, oh, we don't believe it, Spirulakis was crazy, you guys should stop working on this. And then we got, after we'd sort of kept publishing papers on it, it occurs, but it's probably not that important. It probably only occurs in pathological conditions. And then Steve, in this paper, in Circ Research, goes on to show that actually it's quite important in normal conduction. He used a series of dextrans to expand the perinexus in measurable ways and showed that he could very beautifully calibrate and correlate the changes in conduction to the changes in the perinexus. So So Steve has done some very fine work over the years. This was a paper that came out about 18 months ago in Science. It's from Arjun Deb's group at UCLA, and they sort of ran with some of the ideas that we'd been developing and showed that ephaptic conduction may be important at the infarct border zone where the fibroblasts and myocytes interact. The study involved quite sophisticated use of transgenics, including animals that had been with comprehensive loss of connection expression in the heart. And they speculated, although didn't show, that there was probably a junctional cleft of extracellular space between the fibroblasts and the myocytes at the injury border zone. But as I say, they didn't actually demonstrate it in imaging. And so we're still left with the question of whether the fibroblast myocyte cleft at these scars is perinexal-like or perhaps something different. And you know, there could be more than one type of ephaptic conduction that occurs in the heart. One of the things we've gone on to do is to try and build on our peptides to turn them into prodrugs. This is my grad student, Zach Williams, and an initial stab at this was to try and reduce the size of BADP1. It's about 19 or 20 amino acids long, and so we scanned along it, taking 6 amino acid peptides, and identified a domain that in our various assays appeared to show activity similar to the parent molecule. We were also interested in developing an agonist, something that didn't disrupt adhesion but actually promoted adhesion. And there have been reports that if one dimerizes an antagonist in one of these immunoglobulin disrupting peptides, it actually has agonizing properties. And there have been a few papers on other immunoglobulin molecules such as desmoglein-2 and encadherin with the strategies being successful. And as I'll show you in a second, this seemed to work for us as well. We did some mathematical modeling, modeling of the interactions of our peptides with the adhesion surface, and identified that this hydrophobic pocket here is probably important. Here we can see the short peptide LQ lead associating with the immunoglobulin domain. All right, so what happens in our rhesus assays, the assays in which we measure adhesion between the cells? So the red line here shows over 45 hours or so the effect of adding the BADP1 to these cells, and you can see there's a loss of adhesion at least over the first 30 hours or so. In comparison to the short peptide, we can see that the loss of adhesion is much more dramatic. But most importantly, we took a couple of these dimers of the LQ lead-like sequence and popped them on these cells, and to our delight, we found that they showed agonizing possibilities. We got significant increases in the level of adhesion between the beta-1 overexpressing cells over the full 45-hour time course of the experiment. We've got 2 different flavors of the dimer here. One is a circularized version, and the other one's a linear version. And actually, the linear version slightly outperformed the circularized version. And Matt made an unexpected observation with these peptides. It turns out that the beta-1 subunit has other aspects to its function and life cycle, including this property where it undergoes a regulated intramembrane proteolysis to release a C-terminal fragment, which then migrates to the nucleus and modulates gene expression, including for voltage-gated sodium channel subunits. And Zach found that the BADP1 actually upregulated this signaling pathway. Okay, so far all the work I've described to you provides it provides a kind of indirect indication that there may be another mechanism of conduction going on between myocytes other than gap junctions. And so far I haven't really shown you any direct evidence. And this work is unpublished at the moment, and it's being undertaken by my postdoc, Zalbo Wu, who is a remarkable electrophysiologist. And Zalbo, and this is really hard work, but he's developed a model in which we compare the sodium channel activity in single myocytes to pairs of myocytes with intact intercalated discs. And what we're trying to do here is to reduce the complexity down to the simplest form we can. These are some sort of fairly baseline characterization of the sodium channel activity within these single and myocyte pairs. There's the peak sodium channel activity is about double that in the paired cell and then the single cell, about what we'd expect. The kinetics of sodium channel activation is about the same. The proteins of interest, connexin 43, NAV1.5, beta-1, are all present within the intercalated discs. And also, when we look at the perinexal domains, we find gap junctions, but we also find the perinexal domains. And moreover, when we put on our adhesion-disrupting peptide, we can get significant increases in the width of the perinexus between these cell pairs. So, many of the elements that we're looking for experimental manipulation are in place in the cell pair model. Okay, so now this is probably Zalbo's most important observation. When he does a patch clamp of sodium channel at various membrane depolarizations in single myocytes, he sees pretty much the expected sequence of curves. And this is because, of course, the sodium ions are moving in from the outside to the inside of the cell via channels that are distributed around the cell, possibly more concentrated in the intercalated discs than on the sides of the cell, but nonetheless structurally not particularly not showing particular unexpected electrical activity or distribution of the channels. And if we look at the rate at which the sodium channel activation occurs, in this case he's pulled out what we call the pre-peak curve, which is the curve in red just before the green peak curve. It occurs at around minus 50 millivolts. And if we run a first derivative of this curve, we can see that it's fairly straightforward. There's a single trough and not too much else going on within the curve. Now the situation changes in the myocyte pairs because, as I've mentioned, we now have these junctional sodium channels within the intercalated disc. And so what does he see when he does his patch of these cells? Something very different. When we look at the pre-peak curve, and I know it may not look like a lot, but nonetheless, when we run the first derivative, we see the occurrence of two troughs. And we think that this is the ephaptic signal of the junctional sodium currents. We're calling it, we're kind of having a debate as to what we call it. I'm calling it the intercalated disc signature. Zalbo wants to call it the transactivation signature. So, stay tuned when we actually publish it as to exactly what we call it. But nonetheless, we think it's important, and we think that, it's a direct signal of these junctional sodium currents. Now, when we measure our sodium currents, we're doing it at 25 millimoles. And of course, 25 millimoles is only a fraction of physiological sodium. Physiological sodium, and I'm sure there's some physiologists in the room here, I think it's around 135, 145 millimoles. It's way lower than physiological levels. We have to do that because if the sodium concentration in the bathing solution gets too high, we don't get clean sodium currents from the cells. So here is the characteristic set of curves from a single myocyte, and then here is our curves from a myocyte pair. Now, at this concentration of sodium, when we block the gap junction channels with this molecule here, 2-APB, something interesting happens. Basically, the characteristic intercalated disc signature disappears, and the sequence of depolarization curves activation curves, look more or less like a single myocyte. Now, when Zalbo increases the sodium concentration in the bathing bath just another 10 millimoles and takes it up to 35 millimoles, again, and this is basically the same, this is the same pair of cells, he once again recovers the intercalated disc signature, and you can see this red line here has this two-phase initial activation sequence. And if we run the first derivative, again, we can see our two troughs turning up. So what this has taught us is that extracellular sodium is critical for the activation of these junctional sodium currents. And I think, and I'll go on to show you, we've started to do some mathematical modelling of this, but basically what we think is going on is that with the rise of sodium, there is just an enormous flow of charge into this very tiny volume. Steve Polsing did a back-of-the-envelope calculation. He calculated that the charge at physiological sodium is equivalent to the energy density, well, actually 300 times the energy density of a lightning strike. So, if you're talking about the spark of life, this may well be where it's going on. And of course, what are you going to want to rely on? Seepage through a gap junction or a lightning strike to keep your heart going? I know which one I'd go with every time. So, and as I mentioned, we've done some, we've started some mathematical modelling. We have a collaboration with a very talented mathematical modeler at Ohio State, Seth Weinberg, and he's used a fairly classic electric circuit model and converted it into a math-led model that includes the perinexus, the junctional sodium currents and the lateral sodium currents, etc. And here's some very early data from that modelling process. Here's the case of the 25 millimolar sodium. On the y-axis, we have the width of the perinexus, and really probably the region to concentrate in is between 10 and 30 nanometers, which is what we measure within the normal intercalated disc. And then on the x-axis, we have different levels of gap junction coupling. So we can see here, when gap junctions are blocked, the delay in junctional junctional activation doesn't occur. So at this sodium concentration, it more or less looks as though gap junctions are dominating. Interesting, 25 millimoles is about the sodium concentration found in a hibernating mammal, and so it looks like gap junctions might be important in cardiac activation in the hibernating mammal. But as we start moving up the sodium concentration, you can see here, even at low sodium ion at low gap junction coupling levels, we get the re-emergence of the transjunctional activation, the junctional sodium current activation. So this work is ongoing, but nonetheless, we think that it leads, it further leads to the question of actually how does activation occur within the heart, and it may not involve direct contact. And I just want to close, because I know there's some brain folks in the audience, I just want to close on some observations about electromagnetic fields and electrical activation. And I like this little cartoon here. It shows a couple of magnets moving with respect to each other and the way that they change the shapes of the electric fields as the magnets move apart. And there was a very nice paper from Young Kuchera's group in Switzerland a few years ago now, where he proposed that the perinexus is actually a means for focusing electromagnetic fields around the gap junction so that the flow lines of ions are concentrated where these ion channels are most optimally placed. And here's a paper from a group in California, I believe, where they started talking about how focused electromagnetic fields in the brain may contribute to sentience. And in fact, there's really an emerging literature in the brain about haptic mechanisms. They're thought to underlie brain oscillations such as theta waves. There was a paper just a few weeks ago about their role in the formation of memory engrams, and also a proposal, because of their ability to integrate over large tissue ranges, perhaps it provides a way of addressing the binding problem of consciousness. So, those are probably, big ideas, and, maybe haptic conduction has nothing to do with them, but nonetheless, there's some connection between the heart and the brain there. So with that, I will stop. Again, thank the members of my group and also my funding from the NIH and the various gifts and grants that we have from other organisations and other foundations. Also the Ted and Molly Carr Medical Travel Fund for supporting my and Donna's travel to New Zealand. And I'd be happy to answer any questions. Was it 45?
Right. Is this on? Hello. I think so. Questions? Here we go. Julian's got it. It's for the online people as well. You can shout. You can shout into the mic.
Congratulations, fantastic lecture. Very educational. So I suppose the obvious question is what are the gap junctions doing?
Yeah. Well, Cy kind of teases me, and he says, gap junctions are the best staple that nature ever made. They're basically the closest approximation that you'll find anywhere between any two eukaryotic cells. It's the gap from which the gap junction draws its name is 2 nanometers, and so so I think maybe haptic conduction or the perinexus has taken advantage of the fact that these the cells are so close together. To create these cleft-like domains at their edges that, accumulate other, ion channels and other structures that may be useful to the cell. I think, the other work that we're doing suggests that the gap junction in the heart has very important roles in the injury response. And I didn't go into that here, but I think the sharing of metabolic and other information between cells is probably, an overlooked aspect of their function in the heart. It's a fair question, but I think, they do have a communicating role, but I think it's just more complicated than, they're responsible for the activation, propagation from one cell to the next.
Another question?
Congratulations, Rob. Lovely talk. Really another variant on the same question. I mean, one observation is it just amazes me that the role of gap junctions has been sustained in the face of the evidence that's been there, as you've pointed out, for so long. But do you believe that the gap junctions are also providing some kind of a fail-safe backup mechanism? I buy that. As you know, I buy the theory of what you just explained.
Yeah, no, that's a fair and interesting question, Bruce. There's a couple of schools of thought that maybe, the haptic mechanism and the gap junctional mechanism represent two independent mechanisms, and when one, starts going awry, the other one steps in and backs it up. But the other school of thought is that perhaps they're part of a integrated mechanism that ensures activation moves reliably from one cell to the next. And I must say that I kind of favour the second school of thought because, Mother Nature kind of likes redundancy, but she kind of doesn't either. She works very efficiently, and so I think, we'll have to see. I wouldn't like to speculate, but yeah, interesting question.
Yeah. Thank you.
Do we have any more questions? Well, I think, oh, over there.
Hi, Rob. I'm not a physiologist, so this might be a really dumb question, but do you think there's an issue of directionality with the way that things are set up across either the perinexus or the gap junctions?
Yeah.
Could the gap junctions be a trickle charge to the opposite of the, like reflowing the sodium?
Right. Yeah, that's a very interesting thought, Jackie. I think the ephaptic mechanism is inherently more directional than the gap junction mechanism, because the conduit between the two cells, presumably the ions can move in both directions across that conduit, whereas with the ephaptic mechanism there's definitely a step from the upstream cell to the extracellular space to the downstream cell, which propagates it this one way. But you're introducing the idea that the gap junctions might be part of a circuit that kind of ensures that directionality is
Well, there's enough pool of sodium sitting in the gap, and then the perinexus
right.
shooting it through.
Right. Yeah, that's an interesting thought.
You could reverse your experiments, right? With coupled monocytes to test it.
Do you want to just repeat
So I was just saying you could reverse the experiments where you got the coupled myocytes, so go from one cell to one side and then trigger it from the other direction.
Yeah, possibly, but the way we conceive the experiment is the single patch pipette on either one of the cells, and it doesn't matter which one, basically clamps the lateral sodium channels within the assembly of the two cells. What is not clamped are the junctional sodium currents and the lateral membrane of the downstream cell. And so you can't control what they do. And so it builds in a kind of contradiction within the system as to what's firing first. And we're using the models to try and figure that out. But I don't think that approach would allow us to get at the directionality of the question, because the directionality would still be absolute from where the electrode is to the downstream cell.
I was wondering if you could reverse the electrode.
Yeah, no, so it's in one cell, and then you take it out of that cell and put it in the next.
Yeah, or maybe have a couple of electrodes in one.
Yeah, we've tried that. The amplifiers that we use for the patch setup, cheers, Bruce, just really don't support. We can't get the signal. We'd actually have to spend a lot of money to get another specialised type of amplifier that would allow us to do 2 sodium channel patch electrodes. So at the moment we're kind of stuck with the particular amplifier that we have, and I don't quite understand why we can't use 2 patch electrodes. At least I couldn't explain it to you. If Zalbo was here, he could. But nonetheless, yeah, we've thought about that, and we've tried and have been unsuccessful with our current setup.
All right, we might have to just leave it there, but I'm sure you will all agree with me that some absolutely extraordinary work going on for Rob and the team, and I'd just like everyone to say another big thank you for tonight's information. And we'll just have we'll have a quick 5-minute break, so if you're back here by just after 7:10, that would be great. Thank you very much. Yeah. Well, that content was
a non-noisy water bottle with me. I was like, oh, the ones in the hotel don't make noise.
Ah, yeah, no, nice.
Whereas
I know.
these ones are like,
I know. Yeah. Shockers, aren't they?
yeah.
Yeah.
Whenever you want to go, I'm good.
Yeah. Just wait for people to drift back in. But it's nice you've got an audience here to, keep you. Yeah.
Yeah. And two of the people involved in the work here too.
Oh, that's awesome.
Nice.
Is that you and Lynette?
Lynette and then Odyssey, the second girl.
Oh, yeah.
She's one of my PhD students right now.
Oh, awesome.
Yeah.
Cool. Oh, that's super great. Kia ora. I hope everyone's had time to stretch their legs and have a little bit of a break. So our next rock star of research is Professor Donna Rose Addis. And Donna Rose grew up in South Auckland and is the first Samoan neuroscientist in the world, specialising in neuroimaging, and has been living and working in Canada since 2018. In 2016, Donna Rose was made the youngest ever fellow of the Royal Society of New Zealand, and she's been recognised for her pioneering work in the use of functional brain imaging to study how the brain stores and retrieves memories in healthy people and those suffering from neurological disorders. Donna Rose's work focuses on understanding the relationship between memory and imagination, the ways in which future imagination differs with age, mood and mood disorders such as depression and memory loss as a result of amnesia or aging. And I believe tonight is going to be on the self. So please join me in welcoming Donna Rose so she can tell us more about her fascinating research.
Thanks Sue, for that lovely introduction. Talofa lava. It's really nice to be back here at the University of Auckland to speak to you tonight about some work that actually I do in collaboration with some folks here at the University of Auckland who are here with us tonight. So I'm going to be talking about memory, the relationship between autobiographical memory and the self in dementia, but also in amnesia. And so there's a common portrayal of amnesia or dementia as a loss of the self. And so, of course, we see this a lot in the movie industry in particular, where you have individuals who lose all sense of who they are with memory loss. This is also evident in the scientific literature with quotes such as these. So the Alzheimer's person ceased to be the person he or she once was. Severe Alzheimer's disease involves a separation from the self. Or this idea that amnesia impacts the conscious self. So one amnesic case, Clive Waring, described his experience as it's like being dead. And if we look at philosophical perspectives, we also see this very intimate link between memory and the sense of self. And so John Locke proposed that as far as this consciousness can be extended backwards to any past action or thought, so far reaches the identity of that person. It is the same self now as it was then. And then more recently, Parfit argued that the continuity of the self that we experience as persisting over time emerges from the present self experiencing some direct psychological connection with a moment in the past. And so this can be thought of as actually retrieving a memory of a past experience. And so that led us, and in a very big review of the literature in 2013 that was led by our student Sally Preble, to propose a model between thinking about different aspects of autobiographical memory and the self and how these might relate. And so we started from using William James's distinction between the I self, so the self that experiences the world, and the me self, which is the object of one's experience. And so we can think about this in the present moment. So we can have, We can have the I self in the present moment, and this refers to our kind of internal experiences and our awareness in the current moment. We also have the semantic aspects of self, so the self-concept, and together this gives us what we call synchronic unity. So how it is that we integrate all the different aspects of the self in this one point in time. But then we can take this further and see that we have a sense of self that extends over time. So we can have an experience of the self as persisting in time, and so that's a bit like what Parfit was saying about having this experiential connection with the past or potentially with the future. And then we have semantic forms of continuity as well. So I'm going to be talking about evidence that we have for all of these different aspects of self in this talk, and how they relate to different aspects of memory. So just for those of you who aren't familiar with the terminology of episodic autobiographical memory, here we're referring to our experiences, our memories of particular moments in the past, so particular events. So, maybe thinking about the last time that I gave a lecture in this lecture theatre. Or semantic memory being referring to concepts, schemas, and narratives. And so when we're thinking about that with respect to ourselves, these are usually, thought of as facts about ourselves and our lives. Okay, so my outline of what I'm going to cover today is first going to talk about evidence for the present moment self, the temporal I self, and then the temporal me self, and then I'm going to offer an alternative perspective on this that we've been working on in the last couple years. Okay, so when we're talking about pre-reflective self-experience, we're really talking about a couple of processes, one being agency, so being able to tag different experiences as my own. So for instance, if I have the intention to move my arm and then I move my arm, I can, have that sense of self-control over my own actions. Another aspect of pre-reflective self-experience is body ownership and first-person perspective. So the awareness of my body, that my limbs belong to myself, but some other limb doesn't belong to me, that kind of experience. And oftentimes this is stuff that's just happening in a very kind of automatic or implicit way that we're not really focused on. So Olaf Blanke at the University of Geneva has done some really interesting work using virtual reality to show how you can actually modify one's perspective, and he can actually, in various paradigms, induce out-of-body experiences. So for instance here, somebody's actually standing here, their back is being stroked by a tool, and the camera is recording that, and they actually see that happening in front of them. And so then they actually have an experience of being outside of their body. And the interesting thing is that there's certain regions in the brain's default mode network that really are very much associated with the first person experience as well as body ownership. And one of these, oops, I keep pushing the wrong button here. One of these that's most important is the angular gyrus in the posterior parietal cortex. And so it's in combination, for these out-of-body experiences, we see angular gyrus in combination with premotor and sensory type cortices. So self-awareness really refers to our ability to recognize ourselves in its most basic form, as well as self-referential processing. So being able to see different sorts of, the way we test it is to give people adjectives and to think about, get people to judge how much that refers or describes themselves. It also relates to our awareness of different internal sensations, and this is known as interoception, as well as our ability to introspect on our emotional states, our current thoughts, and to be able to construct a unified representation of all of that current self-experience. So both what's happening, what we're perceiving outside of ourselves, but also what we're perceiving internally. And this relies also on regions in the default mode network, now in the cortical medial surface, so ventromedial and dorsomedial prefrontal cortex, posterior cingulate and retrosplenial cortex, in combination with the insula. And so the insula being highly tuned to interoceptive processing, but then also integrating internal and external sources of information. So according to our model then, pre-reflective experience and self-awareness shouldn't be reliant on any of these forms of memory. So this is something that's it forms the foundation, I guess, if you like, of the other aspects of self that our memory contribute to, but in of itself, it should be preserved even when memory is impaired. And so to look at this, I think this might have been one of the first studies to really just actually take a look at this. Sally, in her PhD work, had a healthy adult group. So we had 25 healthy controls, mean age 82, and an Alzheimer's group, mean age 77. And of course, the individuals with Alzheimer's disease were significantly lower in their mini mental state examination, and then also in controlled oral word association, so in verbal fluency. And so in this task, what Sally did was when she first started interviewing them, she took a photo of them. She uploaded that into her laptop, and then later in the testing session, about 45 minutes later, she gave the individual 3 photos and asked them, do you recognize anyone in this photo? If they said yes, they then had to pick out who they recognized and were asked who that was. And 14 out of 15 individuals with Alzheimer's disease recognise their own self. So at a very basic level then we can say they have a preserved sense of self-recognition. The next kind of aspect was to look at the self-awareness. So asking individuals to tell us about anything that they're perceiving in the external environment at that moment, but also their internal sensations, emotions, and thoughts as well. And we had to take account here for the fact that these individuals, given their dementia, have reduced verbal fluency, and so are just then less able to articulate their experiences. So when we statistically controlled for that, we see no difference between the healthy controls and the Alzheimer's patients in the lighter blue in terms of the external information and the internal information that they were able to produce. And on the whole, everybody produces less information about what they're experiencing internally. Okay. All right, so turning next to the present moment me self, so the self-concept. This is also referred to as our self-schema or conceptual knowledge about our current selves. This includes a variety of information, traits, physical attributes, self-evaluations, one's preferences, things that they engage in. It is considered distinct from semantic memory. Louis Renaud and colleagues put forward this model, really distinguishing between semantic memory supported by more posterior, inferior temporal cortices, and then conceptual self-knowledge being supported by more anterior temporal lobe and the medial prefrontal cortex and medial parietal cortices. And so our model argues that semantic autobiographical memory should be sufficient for supporting the self-concept. So in individuals who might have memory loss in other domains, such as episodic memory, they should still have a fairly normal sense of their current self-concept. And so a really interesting opportunity is presented with patients who have hippocampal amnesia. So they have a differential deficit in episodic autobiographical memory. So generally their semantic memory is relatively well preserved. And so it gives us a chance to see, even in this case where they have significant disruptions to memory, do they still have a normal sense of self-concept? And so we had the opportunity a few years ago to test 3 amnesic patients in Toronto. So Odyssey, my PhD student here, came over to Toronto to test these individuals. The person who you're seeing here is patient BL. And so he experienced hypoxia and ischemia. Following a cardiac arrest, he experienced electrocution at his work site at age 24. Interestingly, he has very selective lesions in the dentate gyrus. Oh, I keep pushing this wrong thing here. So here's a zoom in on the dentate gyrus, and you just see that there's some atrophy in here relative to a healthy dentate gyrus there. And these were bilateral lesions. And the next patient that we tested was patient DA. And so he experienced herpes simplex encephalitis at age 42 and had very extensive damage of the hippocampus. So you can see here it's very, very damaged, and other areas of the medial temporal lobes. We also had a third patient, patient DG, who cannot be scanned due to a pacemaker. But he also experienced hypoxia, and given his pattern of deficits, so where he has severe episodic memory deficits and a relatively preserved semantic memory, and we know that hypoxia, that the hippocampus is very sensitive to hypoxia, we can pretty much presume that he also has hippocampal damage. And so Odyssey, with these participants, had them do firstly a trait self-descriptiveness task. And so here what we get individuals to do is to go through a whole lot of traits, rate them for how well they describe themselves, and then we get someone very close to that individual to do the same thing, and then we see how accurate the participants' ratings of their self-concept are to control participants. And we found in that case that all cases showed very similar accuracy to the control participants, or their relative who helped them. They showed similar accuracy, and that was to the same degree as control participants did to their spouse or relative. Another task that we started using actually in my master's research was called the 20 statements test. And this is a task where we simply ask individuals to answer the question, who am I, in up to 20 statements. And so in this case, B, L, and DA generated a very similar number of statements as controls. DG did show a reduction in his statements. I mean, we should kind of potentially note that he does have some small decreases in semantic memory function, so whether that plays a role or not, we're not sure, but it is largely consistent with the hypothesis that the self-concept is not dependent on episodic memory, and that semantic memory appears to be sufficient to support it. So Alzheimer's disease provides a different model for us in that now we have varying levels of disruption of episodic and semantic autobiographical memory. And so then we can look at how these varying levels might correlate with performance on these types of tasks. And so using a trait self-descriptiveness task, here it was just the individuals themselves making these ratings. We didn't have informant ratings. But what was very interesting was that the more severe the Alzheimer's disease, the more of the very kind of extreme ends of the scale the person tended to use. So they would say, I'm always like this, or I'm never like this. And so it was almost like there was a loss of nuance or sort of understanding that maybe sometimes in some situations I might act in one way, but maybe in another way, in another context. And interestingly, this correlated with the loss of remote semantic autobiographical memory. We also had these individuals perform the 20 statements task as well. And so overall, these individuals generate far fewer statements. And the strength of the self-concept is associated with a loss of both episodic and autobiographical memory. But what was really interesting here was that the reduced specificity that individuals with Alzheimer's disease showed. So they were less able to give qualifying details around their statements. So they might say something like, I am kind, but they never give other information about, again, the kind of context or the situations or the people that this might play out in. Whereas controls do that a lot. So they're always giving a lot of extra information and kind of qualifiers. And so this reduced specificity was associated with a loss of episodic autobiographical memory. So in this task here, we're seeing definitely a role of episodic memory coming in. So overall then, this evidence suggests that while semantic autobiographical memory is clearly critical for the self-concept, or is, sufficient to be supporting the self-concept, the episodic autobiographical memory is certainly providing certain levels of nuance or particular types of information that maybe supplement or embellish our understanding of ourselves. Okay, so now I'm going to turn to talking about the temporal I self. And so here we're really referring to one's ability to experience the self as existing over time, both into the past and into the future. And so this presupposes that one has an I self in the current moment, so that one can experience themselves and have self-awareness in the current moment, but that from there they can start to project themselves back into the past and the future, something that's been referred to in the literature as mental time travel. And so a lot of my work has focused on how it is that we can engage in different kinds of episodic simulations that allow us to link discrete moments of self-aware experience. And so you can think of this as kind of traversing the subjective timeline of one's lifespan. And so we have done a lot of work looking at how it is that people can re-experience the past, so really put themselves back into that moment and re-experience everything, about what they sensed at that time, what they, internally, what they experienced externally, and really relive that past moment. And as some have argued, kind of have autonoetic consciousness, so it's a real connection between one's current self and that past consciousness. And then in other aspects of my work, I've looked at how individuals can do this into the future as well. We can do this for both near and remote events. And then over the past few years, there's been a lot of proliferation of literature looking at different kinds of simulations people engage in. One is counterfactual simulations, so how the present moment me can think back to a past experience, maybe where something didn't quite work out how I wished it to. I can rework that experience, hopefully then with the view that if I experience the same thing later, I can change my behaviors or change the situation so that I can get a better outcome. We can also engage in something called retrospective future thinking. So this is kind of new on the scene in terms of something being documented, but we actually noticed this during COVID. When we were collecting data during COVID, a lot of people talked about the fact that their future selves were going to look back at this time and think something about that. So, when I am telling my children about this 30 years from now, I'm going to look back and be like, oh my God, I can't believe we lived through that, or whatever. So and it was very common. So we had these very unconstrained tasks where we collected data during COVID and just had people tell us their thoughts, essentially. And it was really interesting how often that kind of thinking came up. And, most of the time we're doing experiments that are very constrained to things that we, know to be of interest in the literature. And so sometimes it's kind of interesting to just go out there and collect data about, how people think in an everyday, unconstrained way and see what you get back. So that's been a fascinating thing. And then, well, in a way, that is also how we came to start looking at another form of future thinking, so after death and intergenerational future thinking. And so this really, I was saying last night, arose from experiences with my grandmother. When she got over 100, she started having a lot of future thinking around what she was going to do after she died. She had very strong faith, and so she was looking forward to reuniting with her sisters who had passed on. But also a lot of people think very specifically about what people in their family will be doing after they pass away and in their future generations. So another way in which we can kind of have these connections and cross-culturally, I think this is an important form of future thinking that we haven't really explored yet. Okay, and so critical to all of these forms of what we would call episodic simulation, so where you're really constructing these types of episodes mentally, we see the default mode network engaged. Here's activity when people are remembering the past, when they imagine the future, so using very similar structures in the default mode network. And common to both of these is activity in the hippocampus. So what does mental time travel look like in individuals that have hippocampal amnesia? So we tested a number of years ago a patient case HC, and so she had experienced anoxia at birth, and this had resulted in a 30% reduction in the volume of her hippocampus bilaterally. So we can see here the ventricular space is a lot more exaggerated in HC than we would normally see. And so she, in the studies that I'll talk about, there's another study with her a little later, she was aged between 20 and 30 years during testing. And so when we asked her to think about her past experiences or to imagine future experiences, she can locate a number of experiences, so it's not like that she's densely amnesic and can't remember anything. But what you'll see here is that the number of details that she's able to produce, and I just realized my number scale is gone, but irrespective of that, we can see here are the healthy controls, and she is performing significantly lower than those controls in terms of the number of episodic details that she's able to generate when describing past events. And similarly, when she describes the future, she's nearly at floor in terms of her details. So she might be able to tell you that maybe in future she will do X, but then not give you any detail about that. And we have hypothesized in other work that it's likely that these patients, when they're able to kind of give us a little bit of information, that it's very semantic or schematized. In terms of her own self-reported vividness, we can also see a significant drop in terms of both past and future. So we know then episodic simulation is really critical for mental time travel, particularly when we're trying to have that access to experiential details in a very vivid way. All right, so turning to the temporal me self. So now we're thinking about the self-concept and how this might be temporally extended into the past and the future. And so self-knowledge provides for us a model of ourselves that can be continually updated. So we're constantly refining and adding to this sense of the self-concept. And one way in which we can get a really good sense of ourselves is, and in terms of how we're existing over time, is to compare our present self to a past self or a future self. And so this temporal self-appraisal is a really interesting way then to look at one's temporal self-concept. And there are particular biases that humans show when they think about themselves over time. And so one of these is known as the end of history illusion. And so that is the idea that I have evolved or developed as much as I'm going to be. I am the best version of my current self is the best version of me, and people tend to always show this. And so what we wanted to do with case HC is to see, well, does she show these same self-appraisal biases? And she does. So when we ask people about how much they've changed over the past 5 years versus how much they're going to change in future, healthy controls say they've changed a lot in the past, and they're really not going to change that much going forward. And she doesn't significantly differ from controls in both of these appraisals. Another one is known as the self-development bias, and that's where we sort of denigrate or put down our past selves, and it's kind of this, I guess it's a very Western notion that we should always be continually developing in our lives and becoming a better and better version of ourselves. And again, HC also shows that same development over time, actually even more extremely than do the controls. So here we can say that semantic autobiographical memory is sufficient for at least demonstrating these biases of temporal self-appraisal. Another way that we can look at this is to go back to these trait self-descriptiveness tasks. And so here what we do is we get individuals to rate their past self-concept and have someone who knew them back in that time to do the same thing, and then we can compare the ratings to get a sense of their accuracy. And so this has been used with a number of amnesic patients. So we did the same with our 3 amnesic individuals. And in this, we found that BL and DG, their ratings compared very well with their relative to a similar degree as we would see in controls. DA showed a different pattern, and this was, a little interesting. So he rated his past self as much more negative than his wife did. And so there we're kind of not sure if that's actually an indication of, a loss of accuracy, or whether actually maybe his wife is sort of having a rosy retrospection of how her husband used to be before he had amnesia. So, it does speak to the limitations and the challenges of when you're trying to, interrogate something as internal as the sense of self. But generally, we argue that this is consistent with the hypothesis that with semantic memory, people are able to have a fairly accurate sense of their self-concept over time, at least into the past. And this fits with neuroimaging data that shows that it's regions outside of the medial temporal lobes that tend to be most important for rating the self over time. And so here what we can see is, again, doing one of these trait self-descriptiveness tasks. Individuals in the scanner are rating how well this describes themselves, so how selfish they really are. And you can see here medial prefrontal and rostral anterior cingulate cortex being activated. But once people are starting to rate traits for their past self or their future self, then we start to see more lateral temporal regions engaged. So here we're seeing posterior parietal cortex. Okay, so another way in which we can have this sense of the me self as existing over time is to tell stories about ourselves. And this is a very ubiquitous form of internal dialogue, I guess, or something that we all do. We all, to try and make sense of our lives, to synthesize all of the experiences and all of the things that happen to us over time, we have to be able to come up with coherent narratives about this. And usually people will include a set of, or a selection of important episodic autobiographical memories into this life story. So oftentimes these will be turning points, so pivotal events that kind of take your life in some new direction, or they might be events that are very self-defining to you. It involves a lot more what we would describe as potentially semantic or conceptual forms of autobiographical memory. So having representations of entire lifetime periods. So, the kind of the people and the places that, say, when I worked at the University of Auckland, what was it that my life entailed here? And I can kind of have a schema about what that was like, that we used to come here to the CBR for different events. And these feature very frequently in these life stories. But yeah, really the point is being able to synthesize and distill a huge amount of autobiographical content. It relies on a lot of narrative processes. So we're actually constantly revising this life story so that it maintains its coherence. And we engage in autobiographical reasoning. So this is part of how we come to know ourselves and how we make sense of the things that happen to us. And so one thing that we see in narratives are what we call self-event connections. And so this might be where somebody talks about a particular event and says, well, I've always been scared of dogs because I was attacked by a dog when I was young. So there we're getting some kind of self-event connection around stability, or it might be things around change, so particular events and how that led to some change in the self. And it allows us also to incorporate biographical disruptions. And so there can be events that happen in our lives that really kind of rupture the trajectory that we thought that we were going on. And we have to then do a lot of reasoning to then incorporate that into a coherent life story. And often it involves, kind of reformulating schema about our lives. Another thing that we rely on is actually a different form of schema, so one that's really culturally shared, sometimes called semantic temporal chronologies, sometimes called the cultural life script. And so what this is talking about are the prototypical life events that someone in your culture is expected to have. And so to get this kind of information, researchers will have many people in a particular culture say, okay, here's a baby that's just been born. What are all the events that are going to happen, or the most important events that will happen to that person? And so that provides us with some kind of schema for what should be the structure of our life story and the important milestones. And this is particularly important, we think, when constructing a future life story. And so we're actually just got new funding to start a project looking at this in individuals as they approach major transitions, and they're going to start a new lifetime period. So younger adults, as they get to the end of their undergraduate degree and are going to go into their working lives, how they might be using prototypical schematic information to kind of build that future lifetime period, and whether that actually helps with their psychosocial adjustment once they go through that graduation and into the unknown. And then we see that we're going to also look at this for retirement as well, and so then to see how those things might differ. Okay, so we looked at this, again, this is with Sally Preble and Lynette Tippett, and we looked at this in individuals with mild cognitive impairment and Alzheimer's disease. And so they were asked to tell their life story, and then we had blind raters score the number of self-event connections that were evident in their life stories. And so what we can see here is that with individuals with Alzheimer's disease, they're showing fewer of these self-event connections. I mean, they still show a number of them, but much reduced to the healthy controls. They also show reduced coherence, and so there's a number of types of coherence that we score their narratives for. One of them is causal coherence, so are things kind of logically making sense, thematic coherence, and so here we see that the individuals with Alzheimer's disease are down on both of these forms of coherence. Here we're starting to see a bit of a change in individuals with amnestic mild cognitive impairment. And then we also looked at the evidence that they're using these prototypical life events as maybe a schema to tell their life story. And so, firstly I should say, we look at temporal coherence, and we do see that individuals with Alzheimer's disease are significantly impaired on temporal coherence, but even so, it appears that they're using these prototypical life events in their life story. So there's some kind of temporal structure to it. And so we think this might be important because we would expect that with these changes that are with the life stories, that individuals with Alzheimer's disease would show a reduced diachronic unity. And so this is the integration of the self over time. And so we do something called the self-persistence interview. And so here we ask people, do you believe that you are still the same person as you were in your early 20s? How have you changed since then? And how have you remained the same? And then we ask them to reflect on how it might be that one person can act in many different ways but still be the same person. And in particular, this first question about whether you're the same person now as you were in your 20s. We expected that individuals with Alzheimer's disease having all these changes in their life stories would not have that sense of diachronic unity, not have that sense of being the same person. But they were actually similar to the control group in that 50% of the Alzheimer's individuals indicated that they still felt that they are the same self as they were. And here's an example from this interview. So they're being asked, are they still the same? They say yes, oh yes. I don't know why, I just feel I am the same. And the interviewer asks them to reflect on this a little bit more, and they really emphasize this, I feel I'm the same. And then this was replicated later. So Lynette Tippett had a collaboration with Linda Clare in the UK, and they replicated this in a much larger sample, a very much larger sample. So here, 1,465 individuals, and 65% of the Alzheimer's individuals indicated that they had this sense of self-persistence. And so that kind of leaves us a little bit with, okay, so there's definitely a lot of changes to their autobiographical memory, including this reduced narrative processing. So what is it then that's supporting this feeling of self-continuity? Is it that preserved temporal chronology? Is that enough? So just to have some of these kind of landmark prototypical events in their narrative? Or is there something else going on? And so this kind of took us then in a very different direction. And so this is our alternative perspective that we're just putting into the literature now. And so this is the idea of the persistence of self via non-mnemonic mechanisms, so in ways other than through autobiographical memory. And actually, I think, Lynette, you might have played this in my third year neuroscience class. This video, which was what really got me into wanting to look at this particular question about the self and memory. So complaints of a dutiful daughter, and it's about this woman, I think she might have been a journalist maybe, and she's describing in this documentary the experience of her mother going through dementia. And she says, the revelation to me is that my mother projects a very strong sense of self with no memory. She is absolutely a full person without it. And then her mother describes having this continual warm feeling inside. And it's kind of this thing where, we notice when we interview people, they will often, kind of gesture about feeling the same inside. And so that kind of took us on a different track, partly also, I think we were sort of discussing, what might be the longest signal we have in the body, in our lives, and it is being in our body. And that is the most, we're always in that same body. So Odyssey, for part of her PhD, has been undertaking a systematic review of all the possible non-mnemonic mechanisms that might be out there that could underlie the sense of experiential or subjective continuity. And so found a lot of potential mechanisms here. Some we predicted, some we didn't. So, of course, autobiographical memory is there because that's where most of the literature is focused. There was biographical disruptions, or we should really rephrase that as having a stable trajectory, so that when one has a biographical disruption, it shows its effects in terms of disrupting that sense of continuity. What we had predicted through this kind of thinking about physicality was embodiment and interoception. And then we also see nostalgia. So there, it's not about necessarily remembering, but it's about an emotional connection to the past, whether or not you can really remember the details. And temporal processing is another aspect that's come out of this. And so turning to interoception, I had mentioned earlier about this being a really important part of the present moment self. But now what we're thinking about is how it might be that these bodily feelings can provide us with a longer time signals. And so there certainly is evidence about continuity over very short time frames, so over seconds or minutes. that give us a sense of bodily constancy. So here we can just be thinking about our heart rate. We're constantly in a body that has a beating heart. We have things like digestion going on. So all of these physiological processes that give us a sense of being in the same self. And there's some discussion around the fact that we, many of these processes, of course, are regulated to have a restricted range of fluctuation that also gives us then a sense of a similar experience over time. In terms of embodiment, here we're really referring to the history of the body's lived experiences and other bodily representations that we have. And how we might be able to reinstate certain bodily states, perhaps if we're in a certain environment, so maybe coming to give a lecture, that I have the same, internal experiences that I had the last time I gave a lecture. And so there we're getting some kind of reinstatement and continuity of the self, whether or not we're really necessarily aware of that. And habits are another way in which we see actions or bodily kinds of, habits. It's kind of hard to come up with another word for that. But how we engage in these behaviors, let's say, repeated across time, but also across different contexts. And they're very slow to change, which indicates their stability. Finally, temporal processing was another one of these things I'm going to touch on tonight. And so this is basically relates to our time horizons, so how it is that we experience the past behind us and the future in front of us, and how we also perceive the passing of time. And so this in particular can be altered in certain psychiatric conditions, in schizophrenia and depersonalization, conditions like that, where sometimes time can feel like it is passing excruciatingly slowly, or it can actually speed up. Also, how close we feel to the past or the future. This can change, so it can change with age, how we kind of chunk time together once we've lived a longer time span. Interestingly, we actually found this change in the pandemic as well. So people's time horizons really contracted in on them during the pandemic. So that was an interesting finding. But actually thinking about temporal processing made us also reflect on work in the memory literature that is really looking a lot recently at how it is we build a model of the extended present. So the present isn't just the 1 second that just passed. It is usually extended over a period of time. It has a fading sense of the past, as well as some predictions about the future, and we build a representation of this that kind of is a sliding window as we go through time. And so within this, we have overlapping trajectories of various, here mainly internal states, but also external states. So, being in a particular environment, you might feel flushed, anxious, hungry, you might then remember something, and these are all overlapped with each other. And so those overlapping trajectories may also give a sense of continuity. And it is interesting to note that this kind of building up of this sort of extended present is supported by default mode network regions outside of the hippocampus. And so interestingly, in some people, even with severe amnesia, they can build a mental model of the extended present, even if this doesn't get encoded into long-term memory. And so there's, examples of, Brad Postel was talking about interviewing patient HM, and so he would, be able to hold a conversation with him while he was in the room, but if he left the room and came back in, then he didn't know who this was anymore, and everything reset. So that model of the extended present was lost once it shifted, but while it was still there, maybe that is sufficient to give somebody a sense of having a persisting self. Okay, so to conclude, we have, I guess, uncovered very complex relationships between autobiographical memory and the self. So the present moment self is generally preserved with episodic memory loss in amnesia and dementia. The temporal I self relies on episodic autobiographical memory to give us these experiential connections with our past and future, and these are supported by the default mode network, in particular the hippocampus. The temporal me self, so our temporally extended self-concept, is generally preserved in hippocampal damage, supported by other areas of the default mode network and by semantic memory. And life stories are affected in Alzheimer's disease, but diachronic unity is preserved in many individuals. And this has led to us exploring the persistence of self via non-mnemonic mechanisms, including interoception, embodiment, temporal processing, and the flow of experience. And we think that this might be important, that there's this critical level of redundancy, so that even when one is losing their autobiographical memories, the self is really lost. And so I just want to first thank Lynette Tippett, who's here this evening, who really was my master's supervisor in starting this work, and we've now been working on it for 26 years. Yeah. Sally Preble, who was our fantastic PhD student a few years ago, 12 years ago. Odyssey Davis, who's also here this evening as well, who's really spearheaded these new directions. Julia Halalova and Donna Kwan, who work with Shana Rosenbaum, who have done a lot of the amnesic work with us, and Gourdie Pershad, who is doing the intergenerational and after-death future thinking with me. Also, thanks to all my sources of funding, both here and in Canada, and to AMRF and the Ted and Molly Carr Medical Travel Fund for bringing us here. So thank you so much.
Thank you, Donna Rose, and amazing when you say 26 years of work.
That's a good collaboration.
Yeah, totally, absolutely, yeah. But the past is definitely informing the present,
absolutely.
That's for sure. Do we have questions of Donna Rose? Yep.
Hi, excellent.
Have the mic.
Thanks a lot. I was just wondering, do people with dissociative identity disorder or schizophrenia show less diachronic unity?
I'm not sure off the top of my head whether they show less diachronic unity, but they definitely have differences in self-continuity, so in that kind of experience of the self over time. Actually, I think they, certainly with schizophrenia, you also see disruptions in the present moment experiential self. So even, through agency, so you start to see breakdowns in being able to tag experiences as belonging to me. And so then that's partly, what might be happening when individuals have hallucinations. So they're experiencing something, or they're, moving their body in some way, but then they're not tagging that as something that they've enacted. And so then it feels like someone's controlling them. So you get all these different kinds of disruptions of things that are so fundamental to us that usually we're not, even really thinking about that. Yeah.
Hi. I was just wondering, so when you're talking about people who sort of had that loss of ability to think about the future self,
Yeah.
For those individuals, do you also see a loss in the ability to sort of come up with non-self fiction as well, or does that seem to be a completely independent process?
Yeah, that's a really interesting question. So there is research looking at that, and they are able to do that. So they're able to think about, so this was in an amnesic patient that Stan Klein at UCSB had studied, patient DB, and they had him do, a kind of It was actually more, not for other people, but it was actually for the world. So like, what are going to be future concerns or future issues that we're going to have to deal with? And this individual was able to come up with those. Same for the past, but could not do any of this for himself. So it really was a self-related deficit. And so I think that's some of the interesting things around, even when I was talking about semantic, one's self-concept, that is very different from other kinds of factual information we have. So you can have, there are people that have deficits just in self-factual information, in the self-concept, and not in facts about the world. So it's, very interesting dissociations.
Very enjoyable talk, Donna. I was just wondering about your thoughts and reflections on the impact of culture and how these models of self kind of play out in more collective cultures, perhaps.
Mm-hmm. Yeah, I think that's a really interesting question, and something that we haven't really delved into very much yet, but we really need to. So actually, in that future after death future thinking study, the reason we got into that was because my student Gourdie is Hindi, and she was telling me about how, well, she didn't really have to worry about too much stuff because she would just have another life in which she could do these other things. And so then we started thinking, well, we need to really kind of look into this a little more and look at even future time horizons and how they differ, because all of the literature kind of takes this there's a very strong theory around socio-emotional selectivity theory. So this idea that as you get older and your time is limited, you start to shift all your priorities and you prioritize your emotional well-being. So this idea that like people, as they get older, they're more positive, but they're doing this because they have less time left. And that's a very Western concept, right? That your time is going to end. Your time is, and going back the other way as well, your time starts when you're born and not before you. And so that's when we decided to start having a look at this. And so we found a directly opposite correlation. So when we looked at the strength of what's called annihilationism, so the idea that my life ends when I die, the strength of that is negatively correlated with how much time one thinks they have left. So you're more strongly believing in that, you feel like you have less time left, even when we control for age. On the other side, people who believe in spiritual embodiment or reincarnation, they show completely the opposite correlation. So the more strongly they believe in that, the more time that they see as having left. And so that's because we think, and so now we're starting to actually interview or collect narratives from people around this, but I think it's this extension or transcendence of their temporal horizons past their expected death. And so I think this is an interesting thing, because then going back the other way, and I was talking to some Pacifica colleagues yesterday from AUT, and we were talking about the idea that in Polynesian cultures, of course, looking backwards in time is actually the direction that you should be looking. So the idea that you walk backwards into the future because you need to keep your eyes on what has come before you and those lessons and learnings from your ancestors. So I think this is a very interesting thing, and there's a lot of work that we could be doing to look at that. So thanks for asking about that. That's a good question. Yeah.
Yeah, I was kind of interested in that culture question as well.
Mm-hmm.
And I'm sort of and I'm a lay person with respect to this, but I was thinking about the I self, as you were describing it, as a kind of the subjective experience of being me, and the me self, the kind of objective experience of me, my traits, the things I view about myself and so on. Now, is that kind of a dichotomy, or, is there porosity between the two of them? And I'm wondering, with respect to culture, whether that might be the case. And, you know, you mentioned, Asian cultures, but I'm wondering about more primitive cultures like hunter-gatherers or, nomadic herders or, different types of human existence that are, quite distinct from our Western individualistic...
That's right.
Yeah.
Yeah.
So,
yeah, I mean, we definitely know in terms of, cognitive processes that there's certainly, if we just look at something like perception, there's huge differences in terms of cultural, whether you're focusing on details or whether you're focusing on kind of the broader concepts or a broader picture. And we definitely see those kinds of influences when individuals are recounting their past experiences or imagining future experiences. We see all, a very different pattern of what kind of is important and what people kind of focus on. But also another interesting thing, something I didn't really talk about tonight, but that, in Western culture, we really prioritize fine details and having, we talk a lot about specific moments in the past and that we should be kind of thinking about how it is we project back to those specific moments or forwards into specific moments. That's a very Western notion as well. And so when we look at individuals from East Asian cultures as the one that people have researched, they don't have as much of a focus on specific moments. It's more like repeated experiences. So things that happen, the regularities in experience are what are weighted as more important than the unique moments. And so that could also tell us something about, the persistence of self, I think, over time, just simply because you're looking at different time scales there. You're integrating over, 10, 20 years versus, thinking back to one particular moment. So there could be, differences in that kind of way as well. Yeah.
Thank you. Great talk, by the way.
Thanks.
Okay, well, I think we might do a little bit of a wrap now. So I might just ask Robert to come up the front as well, so we can just end the evening on a celebratory note. So I'm sure everyone here tonight and everybody who has joined us online will agree that tonight's talks were absolutely exemplary in terms of standard, and I'm sure we all learnt a lot, and you'll probably go away with some burning questions as well. So please join us tomorrow night, because tomorrow night, something I prepared earlier, we will have Robert talking about milk nanovasculars in the treatment of heart disease and radiation injury, and Donna Rose will be talking about the prospective brain in depression. So there's going to be lots to learn tomorrow night as well, but I'd just like us all to say a huge thank you to both our guest professors tonight for just fascinating and incredible work that you're doing. So thank you very much. Thank you, and we'll look forward to hopefully seeing you tomorrow night as well. Yeah. And do tell your colleagues, we would like to have the auditorium full, please, and online. Get all those people watching. So thank you very much.
All right. Yeah, I just finished reading Joseph Heinrich's book. That's partly what prompted my question.